Cryopreservation and cold chain logistics are among the many challenges the COVID-19 pandemic has forced biopharma to confront head on. While the distribution of deep freeze vaccines has put those issues front and center, maintaining therapeutic integrity through cryopreservation and super cold supply chains have challenged and continue to vex biologics producers of many stripes. These are issues that cell and gene therapeutic developers in particular have wrestled with for years. This is the Business of Biotech. I'm Matt Pillar, your host and chief editor at Bioprocess Online. And today's letter is C, as in cryopreservation, cold chain logistics, cell therapy, and Celyad Oncology. The latter is a clinical stage biopharma focused on the discovery and development of CAR T cell therapies for cancer. Celyad and its head of cell therapy manufacturing, Thomas Lequertier, have openly embraced the cell therapy cryopreservation conundrum. And I'm thrilled he's sitting across from me now to share insight into what his company is doing to ensure the integrity and viability of the important therapies it's producing. Thomas, I know I butchered your last name there. I'll ask you to pronounce it correctly for our audience, but welcome to the show and thanks for joining us.
Thank you, and hello everyone.
Give us the correct pronunciation of that last name.
Yeah, it's Thomas Lequertier, but very complicated French name.
Yeah, we're going from here on out. We're going with Thomas. So your background, Thomas, you worked in production and manufacturing at Dow Corning for quite some time before you joined Celyad, and I'm curious about that. I'm assuming that work at Dow Corning was focused on consumer products, perhaps. And I'm always curious about the transition of career from something other than life sciences to life sciences, and in your case, more specifically, from Dow Corning working in the chemical industry, making a major transformation into life sciences by joining a cell therapy company. So what sort of prompted that move for you? What brought that about?
In fact, there were two main reasons that pushed me to join the CAR T cell therapy field. The first reason is that CAR T cell therapy is a radical improvement for cancer patients. And I strongly desire to participate in the development of that revolution. The second reason is that the manufacturing technology used to make the CAR T is not mature and has to be significantly improved. So that means that situation is quite exciting because in the next decade, there are so many solutions to be developed to properly to have a robust process and so on in the future.
Mm-hmm. Okay. Yeah. Well, so the challenge, it sounds like the challenge was part of the appeal for you.
Exactly. Manufacturing challenge are, there are so many to solve and to address in the future. So it's really exciting.
Yeah. Very cool. What are your responsibilities? So you're cell therapy manufacturing lead or head of cell therapy manufacturing unit at Celyad. What does that entail in terms of your responsibilities there?
In fact, I oversee all GMP and GDP operations of the company, so which include validation and tech transfer, the production, quality control, maintenance, but also the supply chain.
Mm-hmm. Okay. All right. So I'm gonna put you on the spot here and ask you to sort of give us the journey, right? Walk us through the journey, the logistical journey of Celyad's CAR T cell therapies from start to finish. And this is going to kind of lead into this conversation we're going to have around cryopreservation. So, walk me through from start to finish and kind of build in, when and where that cryopreservation process starts.
Yeah. But prior explaining the journey, it's really important to make the distinction between autologous and allogeneic therapies because the logistical journey and challenges are not at all the same. As we know, in allogeneic therapies, the cells come from a healthy donor, so they are ready to go when the patient needs them. But in autologous therapies, each patient T cells have to be engineered individually. And so there are much more challenges in autologous therapies from a logistical perspective.
Right. And Celyad is, so the therapies that you've got in clinic now are autologous. Is that correct?
No, we have both therapies and even more allogenic therapies than autologous therapies.
Mm-hmm. Okay.
And I would say that the journey to manufacture a CAR T therapy can be summarized into 3 steps. In the first step, the cells are collected and shipped to a manufacturing facility. Then they are engineered, cryopreserved, and stored in cryogenic tank. And finally, as soon as the product has been tested and released, we can ship back the product to the clinical site and into a kind of cryogenic shipment box, and we can infuse to the patient.
Mm-hmm. Yeah. So that's the autologous approach.
No, it's the same. It's the same approach. So, except that for allogenic therapy, you have plenty of time to schedule everything. But you also know you also need to collect from an healthy donor, not a patient, but an healthy donor, and so you have plenty of time to schedule all the activities, and it simplify a lot of stuff.
Mm-hmm. Okay, so as far as the cold chain process is concerned, it's much the same regardless. Is that correct?
Yes, it is correct. Yes,
okay.
Except that for autologous therapies, we take the cells from the patient, so they are collected fresh, and you need to start the manufacturing as soon as possible. But in allogenic therapies, you can directly collect. You collect the cells from the donor, but you can already receive it cryopreserved. And so you can save a lot of time, and from a logistic perspective, it's much more simpler.
Mm-hmm. So in your case at Celyad, what is your, describe for me your company's role in the integrity and monitoring of temperature, right? So the temperature control, what temperature controls responsibilities do you bear?
Yeah, in fact, you have to select really a critical step is to select a courier service which is really reliable, because as you mentioned, so the temperature is so critical in the logistical journey, because the cells are very sensitive. And so that means that you need to monitor the temperature all along the shipment.
Right. And how is that accomplished? Are we talking like, what is the actual sort of mechanism that monitors that temperature?
Yeah. So you use a special shipment box, and in those shipment box, you put a kind of temperature sensor that monitor and track the temperature all along the shipment.
Mm-hmm. Okay. What is tell me about, so that part of the process, and we're going to get into the actual cryopreservation here shortly, but that part of the cold chain process where you're monitoring using a sensor, the temperature within that box, is that regulated? Is Celyad subject to some level of regulation specific to that part of the logistical journey?
Yes, it's really temperature is really a critical parameter. So you need to properly monitor that, and it's really a part of the release process of the drug product at the end of the production.
Mm-hmm. Okay, and so the box, the sensors, these are specialized pieces of equipment. Do you work with third-party suppliers to acquire those pieces of equipment, those sensors, and if so, can you share who they are?
Yeah, yes, for sure. We are not, I would say, a specialist of temperature sensor. So for sure, we work with external party. I cannot disclose the name, but yes, for sure, we work with specialists. And on the market, there are, I would say, the traditional temperature sensor that can monitor the temperature, but you only see the temperature when you receive the shipment box on site. But now there are new technology where you can really see in live the temperature along the shipment.
Mm-hmm. Yeah. So is Celyad using that technology now, that networked sensor technology?
Yeah, we are working on that process.
Yep. Okay. Yeah. Yeah. So at this point, when the shipment is received, it's actually the clinician who's validating that the temperature is in the correct parameters.
Yeah, if we talk about the drug product at the end, yes, we send that, and there is we send that to the clinical site, and they've the people really on site are trained, are properly trained really to be able to monitor that temperature, to document that temperature into a kind of batch record, if you want, and they can only use that product if the temperature is still under control.
Mm-hmm. Have there been situations where there's the variance is too large, perhaps, and if so, what then? What happens to that material?
For the moment, we have not yet had that issue, but for sure, if the temperature is auto specification, that means that the cell, the product could not be infused to the patient. And so if it's an autologous therapy, it's not easy at all situation, because there is no other solution for the patient. But fortunately, if it's allogenic therapy, we can send another drug product to be infused for the patient.
Yeah. Okay. So cold chain, is cryopreservation and cold chain logistics a new area for you since you joined Celyad, or did you have some exposure to it when you were at Dow Corning?
So I had experience with traditional cold chain logistics, like the 2 to 8 degree, but I had no experience in cryogenic cold chain, which is very specific to cell therapy.
Mm-hmm. Yeah. Yeah. So drinking from the fire hose, so to speak. That's exactly. It's exciting stuff. Now, so as Celyad advances your pipeline beyond phase 1, obviously, you require more volume. You require the improvement of iterative processes. And cryopreservation is a necessity of that kind of logistical equation, regardless of whether we're talking autologous or allogeneic. But it can also result in significant degradation, right? Loss of living CAR T cells, which diminishes the viability of the therapy. So, and this is the challenge sort of at hand. How do we take the material? How do we preserve it? And how do we maintain its integrity? What is the issue there, Thomas? Why is it that the cryopreservation process results in a degradation of viability?
But yeah, you're right. And so cells are really sensitive, and really during the cryopreservation process, the cells are mechanically damaged by the formation of kind of ice crystals. And those ice crystals can significantly impact the viability of the cells. But however, so the cryopreservation process can be optimized, and so you can play on the for example, on the on the freezing rate. Sorry, you can also add some cryoprotectant. But on the other hand, when you use cryoprotectant, it could be toxic for the cells, and you can also impact the viability of the cells. So it's cryopreservation is really a critical process that you need to masterize to do not impact the efficacy of your drug product at the end.
Mm-hmm. And I know, I've read some material online. I believe Celyad published a white paper. I think it was probably around the time that you joined the company back in 2017, 2018 timeframe. The company's been transparent about the fact that this has been a challenge. I mean, it's not just a challenge for Celyad, it's a challenge for every cell therapy company. Can you give us a little more color on, the process there around manipulation of freeze rates and cryoprotectant, and just what that looks like on the ground level, kind of manipulating those factors and trying to optimize the end result?
But no, there are quite a lot of literature on that. So there are, I would say, most of the players use the same cryoprotectant. And in terms of control rate freezer, you have, I would say, key supplier that can really help you to design your freezing protocol. And when you fix everything, it's really important that that process to be reproducible. So you need to define really critical parameters during that process, like the time, the temperature, the concentration of that cryoprotectant, and so on. And you play on that. And then you fix all those parameters and you reproduce, and you can reproduce that process quite easily without impacting viability.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. Does that, do those parameters change at all or need to be re-equated as volume and scale increase?
In fact, not the protocol. No, the critical parameter are still the same. But so I would say the main challenge is especially with allogenic therapies. The target in allogenic therapies is to produce as much as possible. So right. So that means that you need to identify some equipment that can manage ideally hundreds and hundreds, and even in the future thousands of doses, and cryopreserve those numerous doses. So it's quite a big challenge for the future.
Right. Yeah. And shelf life stability is, I would assume, I would simply assume that it's less of an issue in autologous scenarios than it is in allogeneic. Obviously you want to, as you scale up, you want to build some volume, and you want to maintain the shelf life and stability of those allogeneic therapies in storage. What have you learned? Have you learned, has Celyad gotten to the point now where it's learned about degradation over time, viability over time as allogeneic therapies have sat in cold storage?
No, in fact, the CAR T cells in general are very stable at when they are stored at very, very low temperature. Huh? So really, the challenge relating to the shelf life when you thaw the drug product at the clinical site level. So that means that you are still in contact with that cryoprotectant, and I would say the viability of the cells in contact with that cryoprotectant is only I would say a couple of hours. So that means that all the challenge is really there.
Okay. Describe the financial and sort of expertise requirements around cryopreservation and cold chain logistics. So, obviously it's a specialized step in the process. Does it require specialized expertise and a significant investment in enabling it?
Regarding the investment, so yeah, that's true that an important initial investment is needed, because you need to purchase all the required cryogenic equipment, like the control rate freezer, the cryogenic storage tank, the shipment boxes, as well. And we do not have to forget that all the safety measures to manage to handle the liquid nitrogen. And so it quite, I would say, a quite important investment at initially. And relating to the expertise, in fact, you can have a lot to manage for the cryogenic tanks and so on, the management of all those assets. You can have really the support of the equipment suppliers. But for sure, where you need an expertise is for the cryopreservation process, because it's really included in the manufacturing process of your CAR T, and so it needs to be properly, I would say, controlled, validated, monitored, and it has to be robust. So you need the expertise for that specific step.
Yep. At Celyad, can you give us a glimpse into what team members, what parts of the organization are sort of directly involved in that cryopreservation and cold chain logistics process? I mean, obviously it rolls up, I'm sure, throughout the organization, but who's got a day-to-day hand in it?
But it's as I mentioned, so it's not we've not somebody who is really fully responsible for that. So for the cryopreservation process step, it's I would say our team or validation team or production team with specialized to do that to validate or to do that operation. And then obviously we have infrastructure team to maintain all those utilities assets, and obviously also the logistic teams responsible to manage all the shipments and to make sure that that the temperature is well controlled during the shipment.
Mm-hmm. Okay. What temperature range are we talking about here, Thomas? Give me an idea of how cold we're talking.
The classical specification for the cell therapy, you need to be below minus 130 degrees Celsius.
Mm-hmm. Okay. All right. So a while back, I think it was in February of this year, we enjoyed a conversation with Filippo Petti, your CEO, on the Business of Biotech. Aaron Harris and I, my colleague over at Cell and Gene, had the pleasure of interviewing him. And one of the things that we discussed with Filippo was Celyad's decision to pursue manufacturing in-house. You know, it's always I don't want to say contentious debate, but it's always an interesting conversation with companies like yours about outsourcing versus insourcing. So I'm curious about that decision in the context of the fact that, you know, the cryopreservation is such an important step in the process and a tricky step to get right. Did that play in at all? I mean, I'm sure that decision may have been made pre Thomas, you know, perhaps before you were there. But what do you know about sort of the decision-making process around how the necessity to cryopreserve these therapies plays into whether we outsource or insource? Why not say, hey, there's a whole bunch of CDMOs or CMOs out there who have expertise in this stuff. Let's let them handle it.
Yeah, but really the tricky part, it's in cell therapy, so the cells are very sensitive, and it's not like in the production of classical biologics, when you can at the end of the process you have kind of bulk that you can you can ship afterwards for to cryopreserve or to perform the fill and finish. Here the really cryopreservation is really included in the manufacturing step, because as soon as you add that cryoprotectant, you need to start the cryopreservation step has to be terminated as soon as possible. So that means that if you decide to manufacture your CAR-T therapy, you have to decide to do also the cryopreservation yourself. There is no other option for the moment. And but to be honest, so yes, the cryopreservation is a critical step, but it's far from the most complex of the manufacturing process. So I would say that it does not play a major role into the decision to insource versus outsource that operation, because the other step of to manufacture a CAR-T are more complex.
Yep. Okay. Yeah. So it's a consideration, but certainly far from the determining factor.
Exactly.
Yeah. What is you mentioned the time that it takes to work through the cryopreservation process from the point that you're adding cryoprotectant and preparing to freeze to the point that you're picking and packing the product into boxes for outbound logistical purposes. What does that look like? Are we talking hours? Is it a super slow process? Does it happen quickly?
But yes, so really it's quite. We are talking about a couple of hours, because as I mentioned, as soon as you add that cryoprotectant, the cell viability can be quickly impacted. So that means that you start your final formulation. then you have to fill all the vials of the bags, and then you have to cryopreserve as soon as possible. So that means we are talking really about a couple of hours to do all those operations.
Yep. Okay. Can you, I don't expect very, very specific figures here, but give us a sense for kind of where we were then to where we are now. Celyad's been very transparent, as I said, about its approach to improving and optimizing the cryopreservation process. How far have we come in our ability to maintain viability of these therapies post, through the freeze thaw cycle, versus where we were maybe, 3 or 4 years ago?
But honestly, we do not have any issue. So sometimes, we have to start first, because it's really difficult to compare the cells prior the cryopreservation and after the cryopreservation, because it's really it's very technical, but it's complicated to compare them. But what we can observe is that we do not have any viability issue, and even if you have I would say an impact on the viability. You have to, if you have a really reproducible process, cryopreservation process. Imagine if you have tested in phase 1 your product to patient, and that you demonstrate that that product is safe and with a good efficacy, and you can reproduce the cryopreservation process later, even if you have a small loss of viability during that process. It's not bad, because that you have demonstrated during the clinical phases that your product is safe and the efficacy is good. So if you can reproduce it, you can do it in the future in phase 2, 3, and even commercial stage.
Mm-hmm. Okay. What challenges remain to be solved from your perspective around cryopreservation and cold chain logistics? Where can we still improve?
But I have already mentioned a little bit about that. But with the emergence of allogenic therapies, really the equipment suppliers will have to continue to develop a solution to cryopreserve thousands and thousands of doses in a very short time frame after the end of the fill and finish process. So it's really, I would say, one of the biggest challenge from a manufacturing perspective. But we have also to think about the clinical side, because it's cell therapy, it's still quite new for them also. And so they need really to invest in those cryopreservation equipment. So they need cryotank, they need to properly tow the cell therapies, the cryopreserved cell therapies. So it's also a concern for them, and the challenge is to try to simplify as much as possible the towing process, and also the storage operation at clinical site. Yeah,
that clinical site level control that's intriguing to me too, because it's almost like, if I'm Celyad, there comes a point where I become, perhaps concerned, perhaps anxious, right? Like I want to make sure that this is being handled, to your point, thawed appropriately, administered appropriately. So to what degree is Celyad involved at that clinical site? Like what does your oversight of that process look like?
In fact, it's a very strict process, because they receive obviously training prior handling our product, but after the training, as soon as they have to handle one of our products, they have the kind of batch record, so to document everything they do. So they have to document the temperature, they have to document the shelf when they tow the process, when they infuse the product, and so on. So it's really strict, and it's also really a big challenge, because you have to imagine the clinical side, because I think that we strongly need a kind of industry discussion with the clinical side, because imagine if each manufacturer develop a different kind of towing and managing CAR-T cell therapy of other cell therapy, it will be very, very complicated for the clinical side. So I think that we should regulate a little bit and standardize the practice. So it will be a key aspect for the future.
Mm-hmm. As it stands today in your clinical trials, does Celyad have on-site, sort of feet on the ground at the clinical sites?
No, not at that moment.
Mm-hmm. Okay. All right. Perfect. What do you, if you kind of gazed into your crystal ball, right? I mean, this is your area of expertise now. If you look forward, I don't know, 5, 10, 15, 20 years, what does the future hold? And maybe it's not even cryo. Are there other preservation technologies that the cell and gene space has its eye on? What is sort of the future hold for the ability to preserve, maintain viability, and maintain logistical integrity of cell therapies?
Honestly, I have some. So I think that very low temperature will be always. We will always need a very low temperature. So for sure, the cryopreservation process will evolve, will be optimized after time. But yes, so the cells are living, and so to maintain them during several months or even years, we would need. I think that we would need really a very low temperature to keep a very low temperature, and so that means that. With the potential to cure a serious disease like cancer, the cell and gene therapies market is projected to grow rapidly. So that means that cryopreservation operations are and will continue to be an important aspect of these therapies, and which means that all the players, like the manufacturer, like the courier services, like the clinical site and the equipment supplier, really should continue to invest in the science, in the technology to really improve that cryopreservation process.
Mm-hmm. Excellent. Well, I appreciate this, the time you spent with us, Thomas, and the overview. If you feel as though there's an important question I didn't ask or anything else you wanted to share, now's the time, because we're running short on time. We're going to wrap things up. Is there anything you think being the expert in this space, anything I should have asked you that I didn't?
No, it was really a good set of questions, and we have covered, I think, the most important challenge and aspect of the cryopreservation.
Awesome. Well, thanks again for spending some time with us, and keep up the great work. I look forward to talking with you again. I look forward to talking with Filippo again. I really enjoyed the conversation with him, and Celyad's definitely a cool company to watch. I wish you luck as you move forward.
Thank you.
Thank you. So that's Celyad Oncology, head of cell therapy manufacturing, Thomas Lequertier. Again, I came close with the pronunciation. Yeah. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online, which publishes a fresh biweekly newsletter that I encourage you to subscribe to at bioprocessonline.com. We're produced by Cytiva, which offers, in partnership with Cytiva, which offers a beautiful collection of resources for emerging biotechs on its biotech accelerator, which I encourage you to check out at cytivalifesciences.com/emerging biotech. This pod drops a fresh episode every Monday. And if you like what you heard on it, you really ought to subscribe and give us a solid review. Whether that's Google, Apple, Spotify, it doesn't matter to me. You'll find us wherever you like to listen. Go ahead and give us 5 stars. And in the meantime, thank you for listening.